and welcome to the Oro-Oto-Cerebral Pathway channel. In embryology series today, we are going to talk about the somites. To have a fair bit of understanding of this concept of somites, let us revise a couple of facts. Epiblast is the source of all the three germ layers. All the cartilages, bones, joints, ligaments, muscles are derived from the mesoderm. If establishment of fate map is considered, ultimate fate of cells migrating from the epiblast through the primitive streak, they have been mapped, and the investigations bring about certain results. The cells that ingress through the cranial region of the node will form the notochord. Those migrating at the lateral edges and cranial end of the streak form paraxial mesoderm. Those migrating through mid-streak region form the intermediate mesoderm, whereas those migrating through the caudal part of the streak give rise to lateral plate mesoderm. So we know now that mesoderm is going to be divided into three regions. From midline lateralwards, the regions are paraxial mesoderm, the medialmost, lateral plate mesoderm, the lateralmost, and between those two we have the intermediate mesoderm. Out of these three, the paraxial mesoderm is the one that is involved in formation of the somites. So paraxial mesoderm in prospective head region is unsegmented, where it is the head mesenchyme. Here, to form the mesodermal derivatives of the head region, the head mesenchyme is also contributed by, or tissues to the head mesenchyme are also contributed by the migrating neural crest cells. At the prospective trunk region, this paraxial mesoderm gets segmented, and there we see the somites. The somites are block-like condensations of mesoderm that are located on either side of the neural tube in the developing vertebrate embryo. Somites are precursor population of cells that give rise to important structures associated with the vertebrate body plan and will eventually differentiate into dermis, skeletal muscle, cartilage, tendon, and vertebrae. This process of formation of somites, that is segmentation of the mesoderm in the prospective trunk region, begins somewhere around day 20 in the intraembryonic life. So starting on day 20 in the prospective head trunk junction region, somites start appearing. Every day about 3 to 4 pairs of somite appear in a craniocaudal direction. In the human beings, about 42 to 44 pairs of somites are formed during the entire somatic period, and they range from cranial region up to the embryonic tail. Out of the total formed somite pairs, several caudal ones disappear, and that's the reason why in the human beings ultimately what we have are about 37 pairs of somites. The number of somites that are found is used to determine the embryonic age ranging from day 20 to day 30. So on day 20 we have number of somites present be like 1 to 4. Say around day 25 we will have about 17 to 20 pairs, and on day 30 we have 34 to 37 pairs. So how do these somites contribute in formation of the body plan? The first 4 pairs of somites are in the occipital region, and they contribute in development of occipital part of the skull, bones around nose, eyes and inner ears, extrinsic ocular muscles, and also the muscles of the tongue. The next 8 pairs are in the prospective cervical region. The most cranial ones of these contribute to formation of occipital bone, and the others will form the cervical vertebrae and their associated muscles and other structures. Similarly, next 12 pairs are in thoracic region, and they form the vertebrae, bones of the thoracic wall, muscles and their associated connective tissue elements. Similarly, we have 5 somites in the prospective lumbar region, which will form the lumbar vertebrae, bones of the muscles of the abdominal wall and their associated connective tissue elements, and 5 somites, the succeeding ones, will be in the region of sacrum or sacral region. There they form the sacrum and also form the muscles in this region of pelvis. It is also important to note that the lower cervical and upper thoracic somites migrate into the developing upper limb bud. Similarly, the lower lumbar and upper sacral somites migrate to the region of developing lower limb bud, and that is how we get the bones, joints, muscles, and their associated tendons and other connective tissue structures from the somites, both in upper limb as well as lower limb. So for the upper limbs, we will get them from the lower cervical and upper thoracic somites, whereas for the lower limb, we will get them from the lumbosacral somites. So in the developing vertebrate embryo, the somites split to form, number 1, dermatomes, number 2, myotomes, number 3, syndatomes, and number 4, sclerotomes. Dermatomes are going to form the part of the skin, dermis. Myotomes are going to give rise to skeletal muscles. Syndatomes will form their associated tendons and cartilages, and sclerotomes will actually form the bones. So the dermatome. The dermatome is the dorsal portion of the paraxial mesoderm somite, which gives rise to the dermis of the skin. In the human embryo, it arises in 3rd week of embryogenesis. The dermatomes contribute to skin, fat, and connective tissue of the neck and the trunk. The second component, myotome, is that part of a somite that forms the muscles. Each myotome divides into an apaxial part towards the back and a hypaxial part towards the front. The myoblasts from the hypaxial division form the muscles of thoracic and anterior abdominal walls, whereas the apaxial part, muscle mass, loses its segmental character to form the extensor muscles of the neck and the trunk. The sclerotomes form the vertebrae and rib cartilage and part of the occipital bones, and also contribute in formation of bones of the limbs. Whereas the syndatomes, they will form the associated tendons and other supporting structures related to those bones. And that is how we get the basic skeletal framework of the human body. So the cranial most somites contribute in formation of the head region. Then we have the neck region contributed by the cervical somites. The thoracic somites will form the thoracic skeleton and associated structures of the body wall. The lumbar somites will form the lumbar vertebrae and the structures of the abdomen wall. The sacral somites will form the sacrum and structures in the pelvis. In addition, the cervicothoracic somites contribute in upper limb, and the lumbosacral somites contribute in forming the structures derived from body wall from the lower limb. Well, this is how the somites are formed and give rise to their derivatives. Happy learning!